Hey everyone, it's Jamie Baxter, instructor for Bio 204 AMP2, and we are hopefully going to be wrapping up Chapter 19. There's so many little snippet videos in this chapter because there's so many different topics dealing with cardiovascular physiology. I'm going to apologize. I'm trying to get these videos done because I have a lot going on right now, and I just got back from the gym, so whatever. You're not in my class to stare at a beautiful instructor. You're just getting me straight from the gym. Good thing we don't smell a vision. Let's go ahead and pick up where we left off with chapter 19, and we are moving into the capillary exchange. This is so cool. This part of the chapter is really interesting in how this diffusion through the capillary network works, if that makes sense. So obviously diffusion is the net movement of substances from areas of high concentration to areas of low concentration. And I'm just going to zip back through this. Sorry, I always do this at the beginning of my videos because I just get kind of backtracked. But different substances are going to diffuse differently through our capillaries. And so we're going to take a look at exactly how different molecules go through our capillaries and which types of capillaries function best in different scenarios. So the diffusion is going to depend on a couple different factors. The concentration gradient, what types of molecules are involved, and how far those molecules have to move. So in other words, if there is a very short distance for those molecules to travel, such as just directly, through a small opening or pore, diffusion is going to happen very rapidly. But if the molecules have to go through a series of cells or through a series of passageways, that diffusion is going to be impacted and go much slower. If the concentration gradient is large, or if there's a large difference in concentration gradients on either side of the membrane, that is going to cause rapid diffusion, whereas if the system is almost at equilibrium, there's not going to be much diffusion at all. And then finally, if the molecules are very small, like water, or like sodium and potassium and calcium, just small ions, they're going to diffuse rather quicker than larger molecules like amino acids or glucose or even plasma proteins, which in some cases don't diffuse at all. So this is going to occur continuously across our capillary walls, but depending on the substance, it can occur differently. So there is this chart in your book that goes through everything, but why stare at a chart when we can make a graphic organizer? So boom, that's what we're going to do. We're going to make a graphic organizer of this. So, and at the gym, it was arm day, so my arms are really bad. I'm going to try to do this without screaming in pain because my shoulders are really hurting me. So, diffusion. Maybe I'll scoot my chair over. There we go. Oops, this marker doesn't want to work. Oh well, maybe this one will. There we go. Diffusion through capillaries. So we have something like plasma proteins, which are in the blood, right? These are things like fibrinogen and albumins, right? Most of these are manufactured in the liver. They float around the blood. They help with the osmotic pressure gradients. So these plasma proteins are rather large, right? Proteins are pretty big molecules made of amino acids. Plasma proteins can only get through sinusoids. I know I'm running off the page there. Sinusoids. Sinusoids are the only type of capillary that allow plasma proteins to flow through them. Sinusoids are mostly found in the liver and in the spleen. And sinusoids are, remember, if you recall, I think it was chapter 18, where we talked about the different vessels. Sinusoids have very large holes in them to allow for that. So then we have something like lipid-soluble compounds. Lipid-soluble compounds are things that, such as hormones or even carbon dioxide and oxygen are actually lipid soluble because they're non-polar molecules. So they're lipid soluble, and lipid soluble compounds go directly through the plasma membrane. And that's what I'm writing down here at the bottom of the screen that you can't see. So lipid soluble compounds go through the plasma membrane. I apologize for my bad handwriting. I broke my right wrist, and I'm right-handed about a year and a half ago. And because I just can't get, I don't know, have messy handwriting because of it. Or maybe I'm just lazy. I don't know. So we have large polar molecules. Large polar molecules such as, not proteins here, but this would be something like glucose or amino acids, polar amino acids would be example of large polar molecules. These can only go through fenestrations. Fenestrations are big slits or big holes in the capillary wall. So kind of like a sinusoid, it's a special type of sinusoid called a fenestration. And large polar molecules, like amino acids, can only fit through in those parts. Then we have ions like sodium, potassium, calcium, chlorine, right? All of these can actually just go directly through membrane channels, and we're familiar with this already because we understand the sodium-potassium pump, right? Sodium-potassium pump uses membrane channels. Imagine that. So sodium, potassium, chloride, calcium, major ions that need to shift in concentration in order to maintain homeostasis in the body, use those membrane channels. And finally, water and small ions. I'm trying to think of an example of a small ion. Can't think of any off the top of my head for some reason. It's been a long day. We'll just say water. And this, we'll group the ions in this category because that's the majority of it. But water is mostly just going to go between endothelial cells. Literally between the cells. So you have your capillary. This is our capillary. Right? These are our cells. You see this? Yeah. Between the endothelial cells literally means in between those little cracks. A lipid soluble compound is going to use a plasma protein, and what that means, if we zoom into these cells here, we're going to see that there's little proteins, right? There are little membrane channels and such. Lipid soluble compounds, the sodium, potassium, calcium, etc., they're going to go into the cell, and then they're going to go out of the cell into the interstitial fluids. They're actually going through the endothelial cells themselves, not between them. And they do that by special membrane channels or pumps. The fenestrations would be kind of like this little oops here that I drew, right? You can see that there. I drew like a gap in between the endothelial cells. That would be like a fenestration or a sinusoid, a large space. So let's get back to the lecture here and go back to this. There we go. So this chart here just summarizes exactly what I just drew out. Most of you are visual, so you're welcome. I drew it out for you. All right, let's keep plugging on through. Let's talk about how this capillary exchange happens. I'm going to need to draw again. Oh, I don't have my blanket. I usually use a blanket to erase my little whiteboard. I have a very high-tech setup here in my basement. Either way, while I'm erasing this, I'll explain to you about this filtration process. So as our blood is moving through this capillary bed, there are two things that take place. You have filtration and reabsorption. So think of it like this, right? When the blood arrives at the capillary bed, capillary hydrostatic pressure dominates. Capillary hydrostatic pressure pushes stuff out of the blood, right? So on the arterial side of our capillary, you have the majority of filtration happening. The majority of stuff is getting kicked out of the capillary at and near the arterial side. So it's kind of like you get, imagine those, and maybe, I don't know, maybe you're still in these relationships. I don't know. Imagine you're dating someone, in your early 20s or late teens, and you're living with them. You get your first apartment with them or whatever, and you're in love or whatever. And you have your first big blowout fight, and you just want to kick all their stuff outside, right? And you start throwing their stuff out of the window, right? Get them out or her out, right? That's what's happening here at the beginning of the capillary. Everything gets kicked out, right? And that's because of the capillary hydrostatic pressure, the CHP. Water and small solutes go into that interstitial fluid and out of the blood. This is going to cause what's called the blood colloid osmotic pressure, or BCOP, to increase. And that's because water's leaving the capillary, and now there's all these plasma proteins left behind. So the CHP decreases, the capillary hydrostatic pressure decreases as you move through the capillary, right? There's less and less water in the blood as that blood moves through the capillary, because it's all being filtered out on that arterial end. When this capillary hydrostatic pressure is greater than the blood colloid osmotic pressure, there's going to be a net movement out of the capillary, or pushing all of your boyfriend stuff out onto the lawn, right? But I'm getting ahead of myself. We're going to come back to that analogy in just a second. Remember that analogy. So on this arterial end of the capillary, you also have this idea of net filtration pressure. So net filtration pressure is the difference between the capillary hydrostatic pressure and the blood colloid. Okay, sorry, my